Hello and welcome to a video by Dr. Tinsley to help explain the design investigation report and how to go about doing that for the first practical factors affecting osmosis in potato cells. Now, we'll have gone through the task in some detail in class, but just to remind you, there are four major parts of this: a pilot study in which you practice the method, a research proposal in which you are going to be presenting the design of the experiment that you want to do, the experimental data collection in which you actually will be doing the prac and collecting the data that you need to do, and then part D is your analysis and evaluation, which is really the main part of the written report that you're being assessed on. So those parts will form part of this process, and as you see, you go through, you'll have your submission dates and everything like that. We're going to go through afterwards, looking at now the performance standards for stage 2 biology, and exactly what you need to do for each of those to be working at an A level. So, as this is your first one for stage 2 biology, let's just have a look at, in general, the performance standards. So there is investigation, analysis and evaluation, application, and knowledge and understanding. And these would be the same at stage 1. Within those there are different parts. So investigation parts 1, 2, 3, and 4. As you can see, for this particular practical report, there's no need to address investigation 2, which is looking at acknowledging information from sources. That's much more going to be coming out in an issues investigation. But for the practical report or the design report investigation, what we're really looking at here is 1, 3, and 4. So let's have a little bit of a more detailed look at some of those and how you're going to achieve them at the A standard. So, the first one here is about designing your experiment. Designs a logical, coherent, and detailed biological investigation. So your main job here is really to design the experiment and do it in a way that's at an A level. A large part of this will be happening in your research proposal. So what we have is the research proposal part of that, part B. You'll be looking at this. I'm also going to be looking in quite a lot of detail at your methods here, because your methods show me, method section shows me how that's done in a logical and detailed way. So a lot of those things there will come through in the methods and the research proposal. Let's look at the second part. It's about manipulating apparatus and tools. And at the A level, you're doing that carefully, highly effectively, well organised, safe, with ethical consideration happening. Now this will mostly be what's happening when I'm looking at in-class observations of the pilot study and of the data collection. How well is your bench set up? Are you working carefully? Are you working effectively? Meaning, are you getting the sort of results that we'd expect? And do you clean up afterwards? Are you doing it in a safe way? Proper protective equipment happening? And in biology, we look at the ethical side of things as well, because often we're working with living materials and working with other people, and that needs to be done in an ethical way. Last one here is about your findings. How are you obtaining them, recording them, and displaying them? We're looking for appropriate conventions and formats, and accurately and highly effectively. So where am I looking for this? Well, this is mostly in your results section. And in particular, tables of results and graphs. So, data analysis. Are you presenting in a table? If you're presenting in a table, do you have units? Do you have titles on the headings that on the table that makes it clear? For graphs, there are a number of things to look at. I could spend a whole video talking about graphs, but we're talking about labelled axes, line of best fit, good use of scale, those kind of things there. In the next one, analysis and evaluation. This is one that students find the hardest to do, and this is where I can really tell the difference between A-level students and A+ students. So, critically and systematically analyzes data. Critically. So when you are looking with a critical eye on your data, you're looking for the flaws in your own data and really looking at can you examine and identify those. Systematically means you're working with a system, going through each of those aspects of the data and analysing them individually. Connections with concepts. So people don't often bring this in, but here we're looking at the concept of osmosis. Now we're looking at it in potato cells, but osmosis is something that happens in every cell. can you connect what you find out for potatoes to every cell? Can you connect it to the way an organism operates? Because this is really showing your connections between the practical side of things and concepts in biology. And then what we have here is logical and perceptive conclusions. The logic part means your conclusions have to logically connect to your results. The perceptive is you can see beyond the obvious, see connections and conclusions that are beyond the obvious, and make relevant predictions. So if you find out something about osmosis in potato cells, does this predict what would happen in other plant cells, or in animal cells, or in bacterial cells? Here we're going to look at critically and logically again, but this time in terms of evaluating your procedures. Which procedures were successful? How accurate was your data collection? But most importantly, you need to suggest a range of appropriate improvements to get the A-level grade. So both of these are happening in your discussion. Opportunity to make those connections and analyze and evaluate. Here, you'd be suggesting a range of appropriate improvements for each of those things that you find might not have worked so well in the practical. A clear thing here is we are not talking about mistakes. Correcting mistakes is obvious and low level. What we're talking about is if you look at your method, how could it be improved? Not the way you did the method, but the method itself. Next, in application, we're looking at using biological terms and equations, etc. highly effectively. What are some of the terms you might be using? Now this would be in your whole report. Obviously you need to be talking about osmosis and defining what that is. Often I look for the introduction to see this. You might want to talk about diffusion. In fact, you really probably should. Passive transport, active transport. All these kind of things are part of the biological terms that would be part of this particular report. So make sure you're using them highly effectively, not only knowing what they mean, but using them in appropriate context. This section of application will be again largely judged by in-class observation. You need to show initiative. In constructive and focused, underline that twice, individual and collaborative work skills. How well are you using your class time for your planning, working with others? How well are you using your class time when you're collecting data? Next, knowledge and understanding. So this one is just showing how well you're communicating your knowledge. So this is a big one on communication. So, coherently and highly effectively. So in a report, what types of communication are we looking at? So for instance, the introduction, you need to be able to explain and give background on some information. So there's a large part of written text that's giving background information. Then you have other parts of written text where you're analysing and explaining. So your written text is a key part of this. But we see that there are probably also diagrams that you might be using. There are also going to be tables and graphs. And some of the other formats you might use would come into using quotations or other things like that in other types of reports. But in particular, I'm looking for you to be able to use these different formats to communicate things coherently and highly effectively. Sometimes a diagram is better for explaining something, sometimes text and a diagram work better. So it's all about, are you doing it highly effectively? And that's the end of this video. Good luck, and hope you do your best, and I'll be helping with drafting along the way.